Hello everyone, my name is Vinay Pho and I will be your presenter today. Today's webinar is focused on the BaseScope assay. A few key points to note: you will be muted during this presentation, and please use the chat feature available to you to ask your questions while the presentation is in progress. If we cannot answer your questions during the webinar, we will get back to you offline. As an outline, for the first few minutes of this webinar, I'll be introducing the BaseScope assay, then review the technology, which is very similar to RNAscope. Courtney Anderson, our Senior Application Specialist, will introduce various BaseScope assays, and then I'll discuss the product configuration, workflow, and summary. ACD is pleased to introduce the BaseScope assay, which is a new assay, which is an expansion of the RNAscope technology. This is a novel reagent kit and probe combination for detection of exon junctions, short sequences, and point mutations. The BaseScope platform uses probes that are designed between 1 and 4 ZZs in length for single molecule detection. This is currently a manual fast read assay. The BaseScope assay is an extension of RNAscope technology, and we'll review the RNAscope technology and how it works. RNAscope and BaseScope is based on a proprietary in situ hybridization technology based upon unique probe design and signal amplification. This platform has a sensitivity to detect most genes in situ, as well as simultaneously quantifying multiple RNA transcripts. Just as any in situ assay, RNAscope and BaseScope involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to RNA targets of interest. Specific signal is then amplified using the double Z probe system, which is subsequently detected and quantified. This assay is extremely universal as well as rapid and scalable. To design a specific probe for BaseScope and RNAscope of your target of interest, ACD requests that you provide us with either the accession number information or the exact sequence of the target. You can even indicate your region of interest for targeting with the start or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the melting temperature of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNA conditions. How does the technology work? Probe design is key. We use this double Z probe design, and there are 3 components of the Z. The upper region is the 14-base tail sequence, and that is the region where the pre-amplifier and the cascade of amplifications will occur. A spacer region is in between the base and the other end, and this allows flexibility for the probe binding efficiency to mRNA. The lower end of the Z binds directly to mRNA. This lower region contains 8 to 25 base pair sequences that is specific to your target. These are used in pairs, and in pairs the upper region of the Zs will provide 28 bases for the preamplifier binding. Both Zs bind adjacent to each other to your target region from the lower region. The yellow line represents your RNA. If your RNA is intact under ideal permeabilization conditions, the assay will hybridize up to 20 Z probe pairs to your RNA. But for many samples, such as cancer FFPE samples, your RNA could be degraded or fragmented. The simplicity of this design, relatively short probes bind to the target, allowing for binding even to short pieces of RNA. What our scientists have demonstrated in a lot of experiments is that we just need a few Z pairs to bind to the target in order to detect the signal. Even for fragmented or degraded RNA, which is not likely to have migrated, they're still localized to each other, you should see a single amplification signal for a single molecule. How does the signal amplification work? You should have your target transcript. The probes are added, double Zs land adjacent to each other. The preamplifier will then bind to the top region. Then each amplifier will then serially have 20 amplifiers bind to it. On each amplifier there are 20 labeled probes 20 times. So we see 20 Z pairs with 20 amplifiers and 20 labels, equivalent to 8,000 labels per transcript. That is 8,000 labeled molecules per single target, each roughly 1 kb. What about nonspecific binding? If single Zs bind to a nonspecific RNA, the preamplifiers cannot be stable on just 14 bases of the upper region. So the nonspecific binding that may occur does not get amplified. This is how the double Z design prevents background noise and contributes to specificity. ACD's growth and adoption is best exemplified by the number of peer-reviewed publications. In 2013, we had close to 1 publication per week. In 2014, we saw 84 publications, or 7 per month. In 2015, our total publication exceeded those in 2012, 2013, and 2014 combined. This year, we have already surpassed the total number of publications from last year, with over 200 publications already by September. The images are examples from 8 journal covers from over 15 publications last year. And now I'm going to turn this over to Dr. Courtney Anderson, who's going to introduce BaseScope and its applications.
Okay, thank you very much, Vinay. So now that you've seen the fundamentals of the RNAscope technology, I will focus on the applications of the BaseScope assay. So BaseScope, as Vinay mentioned, is based on the RNAscope technology, but there are several major differences. And here is a general comparison between the two platforms. The RNAscope assay utilizes a probe that consists of typically 20 Z pairs per each RNA target when the RNA target sequence is 1 kb or longer, whereas BaseScope can detect as low as 1 ZZ pair or more. Now, the BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents, and vice versa, RNAscope probes cannot be combined with BaseScope reagents. Both assays are capable of single molecule detection, so that means that a single dot represents one single RNA molecule or target. While RNAscope is a robust assay to detect gene expression of most RNA targets, BaseScope is specifically used for the detection of exon junctions, short sequences, and point mutations, which I will describe in further details in the upcoming slides. Now, as of October 2016, RNAscope is able to detect up to 3 targets simultaneously, whereas the BaseScope is currently singleplex, so it can detect 1 target at a time. And detection with the RNAscope assay can be both chromogenic or fluorescent, whereas currently, again as of October 2016, BaseScope is available with the fast red detection, which can be visualized by either chromogenic or fluorescence. Now, RNAscope can be performed manually or by automation, whereas again currently BaseScope is a manual assay only. Okay. So now I would like to focus on the 3 main areas of application for BaseScope. They are exon junction, short target sequence, and point mutations. BaseScope can also be used to detect targets in certain challenging cases, including targets or samples with compromised RNA quality. Listed in each application area are some specific examples of how the BaseScope assay can be applied, and I'll describe those now in more detail. So first, we will discuss the use of BaseScope to detect exon junctions. Exon junction detection by BaseScope can be applied for the visualization of splice variants, circular RNA, and the confirmation of gene deletion in a knockout sample. These probes are exactly 1 ZZ in length, and they span the exon junction. And I'll show you an example right now. This is the exon junction detection for the variant MET delta 14. This variant is the result of exon 14 in the MET gene being skipped during pre-mRNA splicing. Skipping of exon 14 results in the deletion of the MET juxtamembrane domain and loss of the CBL binding site shown here. Loss of this binding site causes impaired degradation of the MET receptor. So this leads to constitutive activation of the MET receptor. Now, the MET delta 14 variant is present in a variety of cancer types, particularly lung cancer, where it's present in approximately 3% of non-small cell lung carcinoma cases. So detection of alterations in MET exon 14 could aid in identifying patients likely to benefit from MET inhibitors. Therefore, we examine the detection of this variant using BaseScope. Okay. So this is a schematic of the probes used to detect wild type and MET delta 14. Again, all the probes are 1 ZZ in length, and they span the exon junction. And 3 probes were used in parallel. The key probe to detect MET delta 14 spans the exon junction between exons 13 and 15. This probe detects an exon junction that is present only in the MET delta 14 variant. The wild-type probe spans the exon junction between exons 14 and 15. This probe detects an exon junction that's present only in the wild-type MET transcript. And then the last probe spans the exon junction between exons 12 and 13. This probe detects an exon junction that is present in both the wild-type MET transcript as well as the delta 14 transcript. Lastly, we also used 1ZZ probes for the control genes DAB-B, or negative control, and PBIB, or positive control. In general, this can be used as a standard probe design scheme for variant detection, again, in which 3 probes are used in parallel: a probe that detects an exon junction present only in the variant, one that's present only in the wild-type transcript, and a probe that detects an exon junction that's common for both the wild-type and the variant transcript. However, you can consult your account manager and the BaseScope probe design team to confirm the specific design for your experiment. Okay, so now let's look at the data. We tested 3 probes in 2 cell lines. The H596 cell line expresses the MET delta 14 variant, whereas the A549 cell line expresses only the wild-type MET gene. The probe that spans the exon junction between exons 12 and 13 detected signal in both cell lines. This is expected, as this probe detects an exon junction that is present in both the wild-type MET transcript and the MET delta 14 variant. Now, the probe that spans the exon junction between exons 14 and 15 detected signal only in the A549 cell line, and the probe that spans the exon junction between exons 13 and 15 detected signal only in the H596 cell line. And again, this is expected, as this probe detects an exon junction that is present only in the MET delta 14 variant. And lastly, the probe for DAB-B showed no signal, indicating no background signal in either cell line. So taken together, this data demonstrates the ability to specifically detect the MET variant, MET delta 14, in a MET delta 14 positive cell line. All right, so another example of using exon junction is, with BaseScope is the detection of gene knockout. There are 2 strategies that we can do. The first one is, as I mentioned, the exon junction detection. This is for when you know the exact sequence that is being knocked out. So shown here is a highly simplified schematic of gene deletion. A region of the gene that's targeted for deletion by various methods, including CRISPR-Cas9 or traditional mouse genetics. When this targeted region is deleted, there is a rearrangement of the genetic sequence. A BaseScope probe can be designed to detect this newly rearranged junction, referred to here as the knockout probe. We can also design a probe for the wild-type allele, which contains the targeted region, referred to here as the wild-type probe. One would then expect the following results. The wild-type probe would detect signal in the wild-type sample, but not in the knockout sample. Conversely, the knockout probe would detect signal in the knockout sample, but not in the wild-type sample. And this is where the beauty and novelty of BaseScope comes in, because now researchers can see a positive signal in their knockout sample, because BaseScope can detect the rearranged knockout allele, whereas with traditional gene knockout detection methods, the absence of signal suggests that the gene has been deleted. Okay. So a second strategy is to use the short target approach with the BaseScope assay. So if the newly rearranged junction sequence in the knockout is not known, then we recommend you can either use RNAscope or BaseScope. If your detected sequence is greater than 300 base pairs, our standard RNAscope assay may be an easier solution for you. However, if the knockout sequence is shorter than 300 base pairs, a BaseScope probe can be designed for the targeted region being deleted. This probe would detect signal only in the wild-type sample and not in the knockout sample. So the absence of signal would indicate that the gene has been knocked out. Again, you can consult your account manager and the BaseScope probe design team to confirm the specific design for your experiment. Okay. So next, we will discuss the use of BaseScope to detect short target sequences. This is for sequences that are less than 300 nucleotides. BaseScope requires that the total template length be at least 50 nucleotides in order to design a specific probe. Therefore, BaseScope is not applicable for microRNA detection. However, short target sequence detection by BaseScope can be applied for the visualization of CDR sequences for T-cell clones, for which the variable sequence can be less than 20 nucleotides, gene sequences that are highly homologous to other family members, short sequences inserted in gene therapy vectors, small nucleolar RNA, and pre-microRNA, but again, cannot detect microRNA. These probes can be 1 ZZ or more in length, and as an example of the short target sequence detection with BaseScope, I'll discuss the detection of the CDR sequences for TCR clones in Jurkat cells. So the CDR, or complementary determining region, is a highly variable domain in T-cell receptors. To identify the specific CDR sequence expressed in the Jurkat T cell line, we generated BaseScope antisense and sense probes that are specific for CDR3 alpha and beta that are expressed in the Jurkat T cell line. To confirm specificity, we also, as I mentioned, used the sense probes and the antisense probes. We then tested all of these along with control probes, DAP-B and Polar 2A, in the Jurkat cell line with the BaseScope assay. And shown here are the results of this experiment. Antisense probes for CDR3 alpha and beta, which detect the sense or coding RNA sequence, detected strong signal in the Jurkat cell line, whereas the sense probes, which detect the antisense or non-coding RNA, detected little to no signal, confirming the specificity of the Jurkat CDR3 alpha and beta antisense probes in the Jurkat cell line. So now the next application of BaseScope is to detect gene expression in challenging tissues, such as bone or in samples with partially degraded RNAs. BaseScope probe designs for challenging cases can be 1 ZZ or more in length, and they could either be an exon junction or short sequence or a combination of the two probe designs. And this will depend on your specific experiment. Okay, so the first example of a challenging case is the detection of mRNA in bone, which is a classically challenging tissue to detect mRNAs by in situ hybridization. This data comes from the first publication using the BaseScope assay. It came out earlier this year and was performed by Thomas Anderson's group in Denmark. This is the first successful detection of specific mRNAs in specific cell types of human adult bone tissue. The authors used BaseScope to confirm the localized production of TRACP mRNA in osteoclastic cells. They also combined the ISH data with IHC data and demonstrated that the two assays provided different information, but complementary information. The BaseScope assay, shown here on the top, detected TRACP mRNA in osteoclasts only, whereas the IHC detected TRACP not only in the osteoclasts, but also in the reversal cells. So taken together, these data demonstrate that the osteoblastic reversal cells take up TRACP protein that is produced by the neighboring osteoclasts. And finally, we'll discuss the use of BaseScope to detect point mutations. Currently, the detection of point mutations by BaseScope is available only through ACD's pharma assay services and not available as a commercial product. BaseScope probe design for point mutations are 1 ZZ in length. So here we can see the detection of the point mutation EGFR L858R. This is where a single nucleotide change in the EGFR gene results in a change in the amino acid sequence from a leucine to an arginine at the 858 position. Two probes have been designed, one to detect the L858R mutated EGFR sequence, and the other to detect the L858 wild-type EGFR sequence. We also have two cell lines, the H2229, which only expresses wild-type L858, and then the H1975 cell line, which is heterozygous for the L858R mutation. So this means that this cell line expresses both wild-type L858 and the L858R mutation. And as you can see here, with the mutant probe, this probe detects signal only in the mutant cell line and does not detect signal in the wild-type cell line. However, the wild-type probe detects signal in the L858 wild-type cell line, and it also detects signal in the L858R mutant cell line, since, again, this cell line is heterozygous for this mutation. Furthermore, listed on the right are additional mutation target probes that have been validated by ACD, but we are continuously developing more cancer-associated somatic mutation probes. Now, the detection of a point mutation by the BaseScope assay shows how ACD is really pushing the boundary of the in situ hybridization technique. The data shown here demonstrates the current technical capability of detecting even single nucleotide polymorphism. And so in summary, we now have two highly specific RNA ISH assays for different applications. Again, both RNAscope and BaseScope are capable of single molecule detection technology so that every dot visualized is an individual RNA transcript. RNAscope consists of 20 ZZ probes and is intended for standard gene expression, whereas BaseScope is for unique cases with either short sequences, challenging targets or tissues, exon junctions, or point mutations. RNAscope is available as a single or duplex with chromogenic labels and tripleplex with fluorescent labels, whereas BaseScope is currently available as a singleplex with chromogenic label. And lastly, RNAscope is highly sensitive and ideal for most applications. However, BaseScope is ultra-sensitive due to a higher amplification system, and therefore best used for the specific applications I've discussed here. Okay, and now I'd like to turn the talk back to Vinay.
Great, thank you, Courtney. Next, I'm going to talk about the BaseScope workflow and components. Here's a schematic of the BaseScope assay for FFPE tissues. While it's slightly longer than RNAscope by about 30 minutes, the workflow is just as easy. The pretreatment is the same as RNAscope, with hydrogen peroxide to block the endogenous peroxidase, target retrieval to break the cross-links to expose the RNAs to the probe, and the protease to penetrate or permeabilize the sample, followed by probe hybridization. BaseScope preamplifiers and amplifiers are then added, followed by detection with the red chromogen. The BaseScope reagent kit contains several components. Each reagent kit ships with a universal pretreatment kit that contains all the necessary pretreatment reagents. For FFPE tissues, we recommend using protease 3, and for fresh frozen, protease 4 is recommended. The BaseScope reagent kit also contains the BaseScope detection kit with 7 amplifier reagents and the red chromogen. Wash buffer is also included. Please note that the BaseScope reagent kits and probes cannot be interchanged with the RNAscope reagent kits and probes. Since BaseScope target probes can vary in length between 1 ZZ and 4 ZZs, we have designed a control probe pack to suit all BaseScope applications. It is important that you use the appropriate length control probe that matches your target probe. We offer these control probe packs in human and mouse. They contain 4 probes. Each pack contains 2 probes for splice variants, a 1 ZZ probe for PPIB and DAPB, and we have 2 probes for short sequences detection, a 3 ZZ probe for PPIB and a 3 ZZ probe for DAPB. It is recommended that you use this short sequence control probe with all targets that could vary in probe lengths between 2 and 4 ZZs. Polar 2A in lengths of 3 ZZ and 1 ZZ is also available individually. We highly encourage all users to start with BaseScope to start off with the intro packs that contain all the necessary products to get started. We have intro packs in human and mouse, and they contain the reagent kit with pretreatments, the control slide pack, hydrophobic barrier pen, control probe pack, and as well as a vector mounting medium. So in summary, as a result of customer demand, ACD continues with product innovation and now introducing the expansion of the RNAscope assay with the BaseScope platform. This assay is best suited to detect single RNA molecules for short sequences, exon junctions, and point mutations. The assay consists of a new amplification system, probes, and is a manual red kit. To the right, we're showing a subset of short targets and exon junction probes in our catalog, which is growing every day. Please consult your account executives if you're interested in specific BaseScope probes, and he or she will take you through our process of probe design. Lastly, we invite you to visit our support page to learn more. And if you have any questions, please feel free to contact ACD support via email, phone, or via our website. And now I'm going to answer a few questions from our audience. So the first question we have is, can BaseScope reagents be used with RNAscope probes? BaseScope reagents and probes cannot be interchanged with reagents and probes. Another question that's come through: Can BaseScope and RNAscope be run under the same protocol? No. The BaseScope and RNAscope cannot be run together in the same protocol and must be run separately. Can I combine BaseScope and immunohistochemistry? Many customers have performed dual RNAscope and IHC, but dual BaseScope and IHC has yet to be validated in-house. The next question is, can BaseScope detect microRNAs? At the moment, BaseScope nor RNAscope can detect microRNAs. One question is, do we have any catalog mouse probes for the BaseScope assay? Yes, we do have a growing inventory of mouse probes for the BaseScope assay. Please work with your account executive. One question is, are you working on controls for other species of BaseScope, for BaseScope? Our answer is yes. Please do bring forward your species of interest, and we can certainly work with you. Another question is this an RUO assay, and are we working on an IUO? Is there an IUO option? The answer is, this is strictly an RUO assay. One question, is there a BaseScope protocol written for fresh sample tissue, such as cell lines and PBMC? Yes, we do have a BaseScope protocol based off our fresh frozen tissue samples. One question is, can you detect multiple targets at once with BaseScope? At the moment, BaseScope can only detect a single target. Another question is BaseScope available in fluorescence? BaseScope is not available in fluorescence and is only a red chromogenic assay at this moment. Another question is BaseScope automated? BaseScope is not currently automated and is a chromogenic red manual assay at the moment. One question is, can a pool of BaseScope probes, can we use a pool of BaseScope probes? And the answer is yes. Another question is, can you detect circular RNAs? Yes, circular RNAs can be detected with BaseScope.
Another question is, for highly homologous sequences, what's the minimum sequence difference to guarantee the probe specificity? And so while the template needs to be, we need at least 50 nucleotides of sequence for the template. Really the minimum difference is just a single nucleotide. That's all that's needed to guarantee probe specificity. However, if you only have a single nucleotide difference, this would have to be performed by our Farmer Assay Services group, as it would be considered a point mutation. We have another question here. How short of a sequence can BaseScope detect? Again, we can go down to a 50 nucleotide template. If it's more than 50 nucleotides and up to about 300 nucleotides, we can use the BaseScope assay to detect it. So we have another question. If the homology between two genes is less than 300 base pairs, can we use RNAscope 2.5 kit? No, for the RNAscope assay, we really need 300 nucleotides of unique sequence. Okay, so we just noticed some questions coming in through the chat channel here. Is BaseScope better suited to degraded RNA than RNAscope, or are they equivalent? And this may depend on the level of degradation. We recommend you can always start with the RNAscope assay. If you expect that the degradation is severe and that you don't have really short sequences, then the BaseScope assay would be better suited.
Another question is, do you have information on the pricing of BaseScope? We encourage you to follow up with your account executive to discuss pricing for BaseScope and RNAscope. Next question is, will a duplex protocol be available in the future? Yes, BaseScope duplex will be available in the near future, and we'll keep you posted on that. One question is confirming that BaseScope but not RNAscope can detect DNA. I just wanted to clarify that BaseScope does not detect DNA.